Greetings. I am Captain Tsirk. In this video I will address genetic engineering, also called genetic modification, which is the direct human manipulation of an organism's genome using modern DNA technology. The most common form of genetic engineering involves the insertion of new genetic material at an unspecified location in the host genome. This is accomplished by isolating and copying the genetic material of interest using molecular cloning methods to generate a DNA sequence containing the required genetic elements for expression, and then inserting this construct into the host organism. Genetic engineering techniques have been applied in numerous fields including research, biotechnology, and medicine. Genetic engineering alters the genetic makeup of an organism using techniques that introduce heritable material prepared outside the organism, either directly into the host or into a cell that is then fused or hybridized with the host. Genetic engineering does not include traditional animal and plant breeding, in vitro fertilization, mutagenesis and cell fusion techniques that do not use recombinant nucleic acids or a genetically modified organism in the process. Cloning and stem cell research, although not considered genetic engineering, are closely related and genetic engineering can be used within them. If genetic material from another species is added to the host, the resulting organism is called transgenic. First, the gene to be inserted into the genetically modified organism must be chosen and isolated. In animals, the majority of genes used are growth hormone genes. Once chosen, the genes must be isolated. This typically involves multiplying the gene using polymerase chain reaction. If the chosen gene or the donor organism's genome has been well studied, it may be present in a genetic library. If the DNA sequence is known, but no copies of the gene are available, it can be artificially synthesized. Once isolated, the gene is inserted into a bacterial plasmid. The gene to be inserted into the genetically modified organism must be combined with other genetic elements in order for it to work properly. The gene can also be modified at this stage for better expression or effectiveness. The most common form of genetic engineering involves inserting new genetic material randomly within the host genome. DNA is generally inserted into animal cells using microinjection, where it can be injected through the cell's nuclear envelope directly into the nucleus. In biolistics, particles of gold or tungsten are coated with DNA and then shot into young plant cells or plant embryos. Some genetic material will enter the cells and transform them. Another transformation method for plant and animal cells is electroporation. Electroporation involves subjecting the plant or animal cell to an electric shock, which can make the cell membrane permeable to plasmid DNA. In some cases, the electroporated cells will incorporate the DNA into their genome. Organisms are genetically engineered to discover the functions of certain genes. This could be the effect on the phenotype of the organism, where the gene is expressed or what other genes it interacts with. These experiments generally involve loss of function, gain of function, tracking and expression. Transgenics and genetic engineering present intriguing and difficult challenges for 21st century scientists and philosophers. There are a number of ethical questions that need to be considered with genetic engineering. First of all, are we blurring the lines between species by creating transgenic combinations? Also, what are the long-term effects on the environment when transgenics are released in the field? Who will have access to these technologies, and for what purpose? Thank you for watching my video on genetic engineering. Goodbye.